Hi everyone, this is Dr. Han and welcome back to the third lecture in the Biotechnology series. The last time we talked about how PCR is being used as a molecular diagnostic device, but it has its limitation. So in this lecture, we will take a look at how antibodies are being used in diagnostic and biosensing. And we are strictly limiting to our discussion to in vitro use of antibodies in this lecture. So let's get started. Before we talk about how antibodies are being used, we need to first have a brief understanding of the basic principles of various optical diagnostic principles. Then we will look at how antibodies are used in cell-based diagnostics, flow cytometry, antigen-based diagnostics, enzyme-linked immunosorbent assays, and lateral flow assays. The purpose of diagnosis is to detect a disease or an infection at its early stage. If we can catch it early enough, we may have a better chance of treating or curing that disease. Now, modern diagnostics can be divided into two big categories based on either cellular detection or molecular detection of disease biomarkers. For example, flow cytometry can detect different cell morphology and cell surface antigens. Now, we also looked at how PCR is used to detect DNA and RNA markers of pathogens or DNA changes in certain cancers. Now, many diagnostic tests also rely on detecting diseased proteins or proteins associated with certain pathogens. On a large scale, technologies such as DNA arrays can profile a large set of genomic data of an individual to help identify the risk of disease development. So the question is, how can we see the molecular or cellular changes? The answer is using optics. Now, fluorescent signals or changes are most commonly used to detect cellular changes, but it is mostly done with the help of a specialized instrument. Now, on the other hand, if the biomarker signal produces a visible color change, we can use the naked eye to qualitatively tell the presence or absence of a marker, or use an instrument to quantify the particular detected marker as well. Fluorescence is a phenomenon in which a substance absorbs light of a specific wavelength and then emits light of a longer wavelength. Now, this phenomenon occurs when electrons in the substance are excited to a higher energy state by the absorbed light and then return to their original state by releasing energy in the form of light. The fluorescent emission spectrum of a substance is a unique characteristic and that can be used to identify and quantify the substance. Now, when we look at fluorescent signals, there are two terms we need to understand. The first term is excitation. It means when a substance is exposed to light of a specific wavelength, some of the photons in the light are absorbed by the electrons in the substance. This excites the electrons to higher energy levels, and they become temporarily excited or energized. The wavelength of the absorbed light corresponds to the energy difference between the ground state and the excited state of the electrons. The second term is emission. The emission, as the excited electrons relax back to their original energy state, they release energy in the form of light. This process is called emission. And the emitted light has a longer wavelength than the absorbed light. The difference in wavelength between the absorbed and the emitted light is called the Stokes shift. Now, and it is a unique characteristic of each fluorescent molecule. Now, the excitation and emission wavelengths of a fluorescent molecule are determined by its electronic surface structures and the chemical environment in which it is located. And different molecules have different excitation and emission spectra, which can be used to identify and quantify them. In optical emission, the electrons release energy in the form of light. And for fluorescent, the emission is immediate and within nanoseconds. But for phosphorescence, the release of energy is much slower at a much slower time scale. Now, it can take up to several hours. I'm sure as a kid we have all played with objects that contain phosphorescent compounds, such as zinc sulfide and strontium aluminate. Now, besides being used in a toy, optical emission is really useful in helping identify chemical functional units and quantify various molecular species. I have to say that again, in my lab, we routinely use fluorescent dyes to quantify binding affinities between different biomolecules. Now, so far, the fluorescent molecules we talked about were mostly chemical or small molecules, but in fact, biomolecules such as proteins can also have fluorescent properties, such as the green fluorescent protein or GFP. A GFP can emit green fluorescence when exposed to blue or UV range light. It is a very robust protein that can be coupled or linked to another protein. Because GFP is naturally occurring in some jellyfish species, the protein is harmless for most living organisms, in contrast to chemical fluorescent dyes that are toxic. Now, this amazing property of GFP has allowed it to be cloned into other organisms as a non-invasive fluorescent marker to study biomolecule interactions in living animal or in live animals. Now, the technology has opened up so many doors in the research community, and therefore the three scientists who discovered GFP were awarded the 2008 Nobel Prize in Chemistry. As I mentioned, GFP is a non-toxic molecule, and so it can be transfected into cultured cells, cloned into organisms, and fused with another protein using recombinant DNA technology. Other scientists have also mutated the GFP a little bit and create variants that can fluoresce in other colors, such as blue, cyan, yellow, and many more, like a rainbow color. Now, the figures are just a few examples showing fluorescent proteins are being cloned in other animals. I particularly like the beach sunset picture. It was drawn with E. coli cloned with different color fluorescent proteins. I guess this is the ultimate expression of artistic talent from scientists. Another commonly used optical detection technique is colorimetric. Now, first we have enzymatic reactions with the use of horseradish peroxidase. The enzyme can react with a specific substrate to create a color change, and the changes can then be measured with an instrument and or observed with the naked eye. This principle is applied in enzyme-linked immunosorbent assay, which we will discuss in the later part of the lecture. The second less commonly used colorimetric measure is called surface plasmon resonance, or SPR. Now, this property exists in metallic nanoanalyte or nanosphere. Without getting too technical, surface plasmon resonance, or SPR, is a phenomenon that occurs when light interacts with a thin metal film like gold or silver that has been coated on a glass or plastic surface. And when light hits the metal film, it causes the electrons in the metal to start oscillating back and forth, which creates a wave of electrons that travels along the surface of the metal, and it can be detected by a detector. Now, SPR is commonly used in biosensors and other analytical techniques to detect and measure biomolecular interactions or binding of a protein to a small molecule or to other like biomolecules, or such as detecting a virus in a sample. So in my graduate work, in my PhD work, I routinely use SPR as well. Now, it can also be observed in a localized metal nanoparticles, when we call that localized or LSPR. Now, for example, gold nanoparticles with less than 50 nanometer in diameters resonance at around 520 nanometer and can scatter light in the red spectrum. Now, this red spectrum property is readable by the naked eye, and we will see examples of how it's being used in lateral flow assay in the later half of the lecture. And here is a bottle of gold nanoparticles that I used in my lab for developing biosensing assays. Now, most of what we have talked about are detection methods that are used in vitro, now with the exception of GFP that can be applied in live animals. Now, we will talk about how antibodies are being incorporated into those detection methods in a little bit, but now let's switch gears a little bit to look at biotechnologies for cellular diagnostics. Antibodies are often used in cellular diagnostics because they have a high affinity to tag or bind to specific epitopes. We can target both diseased and healthy cellular markers with different antibodies to distinguish the different statuses of the cells. And to quickly provide a readout, antibodies linked with fluorescent dyes can bind to different cell surface marker, and when these cells are subjected to an instrument called flow cytometry, different cell populations are immediately distinguished through different fluorescent tags. So what is flow cytometry? It is an optical diagnostic device that is widely used in both research and clinical laboratories for disease profiling. Now, it measures both the physical and chemical characteristics of cells as they flow in a fluid suspension across an illuminated light path. What is so unique in flow cytometry that makes it so popular in aiding diagnostics? Now, because it can provide multiple readouts for a large number of cells in a very short time. The beauty is that the readout provides information at a single cell level, so it can detect very rare cell populations in a given sample. When flow cytometry is coupled with a cell sorting device, it allows the physical isolation of rare cells that may be of interest. The basic mechanism of a flow cytometry is that it can read live biological samples. Cells in a sample are labeled with fluorescent dyes linked to antibodies that bind to specific molecules on their surface. The sample is then passed through a flow cytometry, and as the sample flows through the machine, it passes through a small nozzle that generates a narrow stream of fluid, and the lasers in the machine then shine lights onto the cells as they pass through the stream, causing them to emit fluorescent light. The machine's sensors then can detect the emit light and measure various properties of the cells, such as their size, shape, and fluorescent intensity. Now, this data is then collected and analyzed using specialized software, allowing researchers and clinicians to identify and characterize different types of cells in the sample. Now, this method allows the quantitative and qualitative analysis of several properties of cell populations from virtually any type of fresh, unfixed tissues or body fluid. Now, the most commonly analyzed materials are blood, bone marrow aspirate, and lymph node suspensions. So how do the cells line up one by one in the machine? Now, once the sample is injected into a stream of sheath fluid within the flow cytometer, they are forced into the center of the stream, forming in a single line, by the principle of hydrodynamic focusing. Now, that means only one cell or particle can pass through the laser beam at a given moment. Light scatter is another one of the properties that can be measured during flow cytometry analysis. Now, when light from a laser interrogates a cell, that cell scatters light in all directions. The scattered light can travel from the interrogated point down a path to a detector. And there are two types of light scatter that are commonly measured in the flow cytometry: forward scatter and side scatter. Forward scattered is the measurement of light that is scattered in the forward direction as the laser beam passes through a cell. Now, this measurement is related to the size of the cell and membrane permeability, with larger cells scattering more light in the forward direction. Side scatter, on the other hand, is the measurement of light that is scattered at a 90-degree angle to the laser beam as it passes through a cell. Now, this measurement is related to the internal complexity or cytosolic structure of the cell. Cells that have more internal complexity or granularity will scatter more light in the side direction. Now, together, the forward scatter and side scatter measurements provide information about the size, shape, and internal complexity of the cell in a sample. And by analyzing these measurements, clinicians and researchers can tell and differentiate the types of cells, such as different immune cell populations, bacteria, or cancer cells. An extension of flow cytometry is another technology called fluorescence activated cell sorting, or FACS. Now, FACS is a technique used to sort individual cells based on their physical and chemical properties. The process involved labeling cells with fluorescent marker that bind to specific molecules or structures on the cell surface or inside the cell. So this step is completely the same as just a regular flow cytometry. Then the labeled cells are suspended in a stream of fluid and pass through a laser beam, and it lights and excites the fluorescent markers and emit light, and all these being detected. Now, the difference is that based on the fluorescent signal, the cells are sorted into different populations using an electrostatic charge, and that charged particle are deflected by electric fields, causing them to be directed into separate collection tubes or bottles. Now, FACS is commonly used in research and clinical settings for a variety of applications, including cell sorting, cell cycle analysis, gene expression profiling, and protein analysis. So what is the real-world applications of analyzing forward scatter and side scatter? Now, the light scattered in the forward direction is proportional to the square of the radius of a sphere, and so to the size of the cells or particle. And studying forward scattering and side scattering allow us to differentiate the different cell types. And we use this method to analyze blood sample. It can quickly identify and quantify different blood cells. The smaller one would be rubber cell debris or even dead cells, and the relative bigger one would be the white cells. And depending on the granules in each of those white cells, we can also finally separate them into granulocytes and agranulocytes. Now, here are some examples of commonly used fluorochrome or fluorescent dyes that emit light from blue-green to red. And in the immunology series, we frequently describe T cells based on their CD types, such as CD4, CD8. Now, because CD molecules are different, we can use specific antibodies with fluorescent dyes to tag each CD. And here is an example of how to use flow cytometry to quantify T cell populations in a given sample very quickly. Now let's come back to look at how diagnostic tests use antibodies to detect specific disease antigens when combined with the optical detection methods we mentioned at the beginning of the lecture. The central principle in any antibody-based antigen diagnostic test is to identify disease-related antigens in vitro biological samples such as blood, urine, and sweat, and many more. Now, because these biological samples carry health information or the status of an individual, the antibodies in the testing kits can recognize free-floating antigens. Now, during my PhD study, I was involved in developing a proof-of-principle assay to detect Clostridium difficile toxin B in human fecal samples. Now, the brown liquid is the real sample. And the chubby hand behind it was me wearing an extra small-sized gloves because we happened to run out of the correct size that day. Now, this work was published as part of my PhD dissertation, and needless to say, the experiments were gross. Another commonly used antigen test is called enzyme-linked immunosorbent assay. Most often people just refer it to as ELISA. Now, it is particularly useful to identify soluble protein analytes and antigens in body fluids. The assay requires the high specificity properties of antigen-binding antibodies. There are several types of ELISA. A direct enzyme-linked immunosorbent assay or direct ELISA is a type of ELISA that is used to detect the presence of a specific antigen in a sample. It is called direct because it directly detects the antigen without the need for a secondary antibody. In contrast, an indirect assay uses a secondary antibodies to target the primary antibody. And why is that? Well, sometimes we just don't have antibodies that can tag with a enzyme that can directly recognize antigen. That's why we need this indirect format. And capture assays or sandwich ELISA is a capture antibody to grab onto the antigen and then measure the binding with primary and secondary antibodies. And likewise, some antigens just won't stick onto the plastic, and we need a capture antibody to grab onto it first so that the primary and secondary antibodies can grab on it subsequently. Now, however, regardless of which type of ELISA, a substrate specific for the enzymes is needed, and usually, specific for an enzyme called horseradish peroxidase is added to the plate. Now, the enzyme catalyzes a reaction that produces a detectable signal, such as a color change, and the signal is measured using a spectrophotometer or other detection system. The amount of signal is proportional to the amount of primary antibody present in the sample. Lateral flow assay or LFA is a simple, rapid, and low-cost diagnostic test used to detect the presence or absence of a target analyte in a sample such as a protein, antibodies, antigens, or even nucleic acids. It is commonly used in point-of-care testing and remote settings where access to laboratory facilities is limited. And it really doesn't need a highly trained person to do the test. And perhaps one of the most utilized LFA is the pregnancy testing kit, which detects pregnancy-related hormones in urine. The basic principle of an LFA is that the sample flows along a strip that contains immobilized capture reagents such as antibodies, and the strip is composed of a porous membrane which separates the different components of the assay. Now, the strip has a sample pad, a conjugate pad, a test line, and a control line, and that's the basic setup. Now, the sample is added to the sample pad, which contains the buffer solution and flows along the strip by capillary action. The sample then reaches the conjugate pad, which contains labeled detection reagents such as gold nanoparticles conjugated with antibodies or other probes. The labeled reagents bind to the target analytes in the sample, forming a complex. The complex then fall along the strip and reach the test line, which contains immobilized capture reagent that recognizes the complex. The complex is captured at the test line, forming a visible colored line indicating a positive result. The control line, which contains immobilized capture reagents that recognize the labeled detection reagents, always produce a visible colored line indicating the test has worked properly. The intensity and the location of the colored line are read and interpreted by the naked eye without the need for specialized equipment or trained personnel. The result can be obtained within minutes, making the LFA an ideal diagnostic tool for rapid and decentralized testing. And I'm sure without me saying, this is by far the most commonly used lateral flow assay since spring 2020. During the early phases, which was around April 2020, an antibody test LFA quickly became available. Now, it could test the presence or absence of IgG and IgM specific to the infection. And from our discussion in the immunology series, we know that the first antibody secreted is IgM, and after isotype switching and successful seroconversion, plasma cells would produce IgG. Now, this was a particularly useful test to identify the infection history of the patient rapidly. Later on, several antigen LFA tests were authorized. Now, some require a machine to provide a readout and some do not, but the overall principle is still the same. Antibodies were used to detect protein antigens specific to the virus. And in a nutshell, samples were collected from the patient's throat or nose, and after mixing the sample with buffer, if the samples contains viral protein, it would be detected with antibodies that are coated on the LFA membrane. Now, take-home tests provide a readout in a few minutes and can be read with the naked eye. And some more specific tests do require a machine to get a more precise readout. Now, overall, we have looked at how antibodies linked to a probe or a tag can be used in diagnostic tests. And in our next lecture, we will explore how antibodies have proven to be useful in treating cancers. So stay tuned, and I will see you next time. Take care.